Okay, so I'm going to get started. So, hi everyone, thank you for coming to my talk today. So, I'm Ben, and today I'm hoping to share with you about the graphical models literature and how we as a community can tap into it to benefit both practitioners and methods developers, especially if you're studying evolution on networks. So, this is joint work with my excellent collaborators, Paul and Cecile. So, Cecile is here, and Paul is not here. Okay, so our starting point is a phylogeny for a set of species. So for example, if you look at the top figure, then you see that we have a tree on the left and a network on the right. So the nodes in the phylogeny, as you've been told over and over again, think of them as species or taxa at some level, and the edges represent descent. And so strictly speaking, all trees are networks, but not all networks are trees, because in networks, we allow for hybrid nodes. So we allow for nodes where they have multiple parents. So for example, if you look at the right figure at the top, X5 is a hybrid node. And hybrid nodes are useful for explicitly modeling gene flow events, such as admixture, migration, introgression, hybridization, horizontal gene transfer, recombination. So I'm interested in modeling trait evolution on a network, and unfortunately, evaluating evolutionary models on larger networks tends to be disproportionately more costly. So for example, if you look at the bottom figure, here I'm plotting characteristics of some empirical and simulated networks. And the x-axis, you think of this as just a naive way of measuring network complexity. We just count the number of hybrid nodes. And the y-axis is, the definition is a little bit more involved, but what you need to think of is this is the cost of model fitting associated with network complexity. So specifically what I mean is that, of course, the cost of doing computations increases as the size of your network increases. If your tree grows larger, you'll spend more time doing the same, trying to achieve the same objective. But we can think of complexity that's disassociated from the size of the network. And so that's kind of how reticulated the phylogeny is. And yeah, okay. So the message is that we can see from the plot. So if you look at the colored points, the darker the points are, the more taxa they have. So we can see that larger networks tend to be more complicated. They tend to have larger X values, and then more complicated networks also tend to be more costly for model fitting. Okay, so here's an example. Suppose we wanted to model how virulence evolves for some pathogen. So if we have a tree with 40 tips, then evaluating a model on this phylogeny is not that costly because trees have low complexity. So this thing that we call max cluster size, that was the y-axis value in the previous slide. That's only 2 for trees, regardless of how large the trees are. But then what we really want is we want accurate models for a variety of reasons, and a tree may not always be the most accurate model if you're dealing with organisms where you have a lot of horizontal transfer or gene flow that's not vertical descent. Right, so recombination and horizontal gene transfer are prevalent in some pathogens, and so the true phylogeny is actually quite reticulated. So you can see here that the cost has jumped from 2 to 54. And yeah, I would say that we need to be able to assess models on larger networks. Currently, a lot of the methods we have, they spit out networks which are simpler in terms of network complexity, but as we're beginning to move towards the direction of generating more complex networks, we need ways to fit trait models on these things. So to facilitate larger studies that account for reticulate evolution, we need efficient methods, and the plug I'm making is that studies and methods at scale are the future. Okay, so this is just a brief overview. So why fitting a model is costly? So our model basically tells us how to compute the probability of observing trait values at the nodes. And so, but we only really observe the tip values. And so what we want to do is to compute the probability of those observations alone. And that's what most of us may be familiar with as a likelihood. And it's how we score different parameter values for our model. So for example, in some cases, we may have exact formulas for getting the best parameters for a given model, but if we don't have exact formulas, then we kind of have to do this iterative process where we try out different parameter values and then calculate this score again and again. But calculating this score involves this summation, and this summation is costly, especially when the ancestral relationships are complicated. So some general strategies that we can take to address this computational bottleneck are to perform the summation cleverly, to have an approximation that scales, that can be computed in an easier, faster way, or to do more clever search in this iterative process. And these are actually all facilitated by the graphical models framework. So so in a nutshell, the graphical models framework starts with identifying direct dependencies on among the variables we are modeling. So for example, in the case of vertical descent, the trait of the species depends directly on that of its parents, and you can model this relationship graphically by a single edge. But in the case of gene flow, the trait of the species may depend on the states of multiple parents, and so we model this as a hybrid family consisting of multiple edges into a child node. So we represent these dependencies jointly on a graph, and then mathematically we also need to characterize how each of these dependencies work, like how does the state of the parent determine the distribution of the child node. And then once we have all this information, then we have generic algorithms actually to process groups of nodes in the graph to build out various quantities of interest. So these are examples of quantities of interest, and a new contribution that we made was actually finding a way to build up the optimal parameter values for the homogeneous Brownian motion model. Okay, so to those of you looking closely, you might recognize that the graphical models approach is not exactly entirely new to the evolution community. So in fact, the well-known pruning algorithm is a special case when your graphical model, this graph of dependencies, is a tree, and what you do is you process sister nodes up the tree and accumulate the likelihood at the root. You can also accumulate the ancestral reconstruction of the root node at the same time. But more generally, when your graphical model is a network, then what we do is we build an auxiliary graph structure called a cluster graph. So in this case, the vertices of the cluster graph are just groups of nodes. And then we do the same thing. We process the vertices in a post order. And this approach is known as belief propagation. So when the cluster graph is a tree, like here, by doing this traversal, we can accumulate the exact likelihood. When the cluster graph contains loops, then what we get is actually an approximation. But allowing for loops allows us to have smaller clusters, and smaller clusters are less computationally costly to process. So that's the trade-off between approximation and scalability. So a natural question is how to find G. Is G always the same as the actual phylogeny? And for simple models, or for most of the Gaussian models we work with, it typically is, but for more complicated processes, it may not be. So generally what you need to take away from this picture is this is the evolution of a biallelic marker within a species network, and we want to take account of incomplete lineage sorting when we're modeling how likely it is that we observed a certain number of red alleles out of all the sampled alleles at the bottoms. And here I'm just showing that model. These are the variables we're interested in: the red allele counts, the total allele counts. This is at a speciation event, and I'm showing you the equivalent graphical model. So incorporating incomplete lineage sorting via the coalescent process is more complicated. It results in a more complicated graphical model. So G depends on both the network and the model. And Right, so far I've talked about an abstract framework, but what are some concrete steps towards implementation? So I'm currently working on software that applies this framework to the continuous trait setting. So continuous traits are widely studied, so I've given some examples here. Our current prototype models continuous trait evolution using linear Gaussian models. So this is the case where the child state depends linearly on the parent states, and then you have some Gaussian noise that's independent of their states. And this basically captures most of the Gaussian trait models that you may be familiar with. Okay, so here we demonstrate our prototype on a really simple example. Here I'm using leaflet data from the genus Polemonium. This is a flowering plant genus with compound leaves that's widespread in North America and Northern Eurasia. It has a broad latitudinal range, and it also has a broad elevational range. So here I'm specifically fitting just a very simple model, univariate Brownian motion on leaflet length along this 17 taxon network that was inferred using SNAC. So here, the three approaches that I mentioned earlier. So this is, the first row is the new approach that we came up with, how to get the optimal parameters for the Brownian motion model. The second would be that iterative process, but using clever summation. And the third would be using that auxiliary graph structure again, but having it be loopy, having cycles and smaller clusters. And so the orange digits, they're just showing you how much they match. And so you can see that the approximate approach can actually it produces good estimates. And this network is too small and simple to accentuate the scalability gains at the moment, but I hope to show that at least this is plausible. Of course, we'll give stronger evidence of how it works on larger cases. Okay, so to recap, networks are needed to model reticulate evolution more accurately, and efficient and scalable methods that apply to larger, more complex networks are needed. The graphical models literature actually offers a flexible framework and a rich toolkit. And then I showed proof of concept, and I just want to emphasize that we need more large-scale studies that start using our tools, and then we can have this feedback cycle between the practitioners and methods developers. And for those who are interested in methods development, I would say that there's a lot of room, even if the graphical models framework is quite well studied, because our pattern of data is somewhat different from traditional applications in engineering, and so that leaves a lot of structure that we can use from our current setting to specialize. And then with that, I will hear some references, and I'll thank you for your attention. Yep.
Super cool. I'm so glad someone's doing this. So when you have those clusters of nodes and you're like processing a cluster of nodes, or are you just like building up a full multivariate normal? Like how do you process like the clusters of nodes in the graph, I guess is my question. Maybe that's too complicated to answer.
Well, okay, so simply each cluster maintains some information, and that information is kind of the local distribution, what's the marginal distribution of all those nodes in the cluster. And as you run the algorithm, these should converge towards the correct marginals. Okay. Right. And then, when you're passing messages and processing information, that's sharing your state so that you agree on the common variables. Yeah.
I think that's me. So good afternoon, everyone. Thanks for coming. I see a lot of you here. So today I'd like to talk to you about what I'm going to assert might be the most important article in phylogenetic comparative methods that you probably never read, or perhaps you've not even heard of it until today, unless, of course, you follow my blog, in which case you've heard a lot about it, particularly over the past few months, including via this post, which has the exact same title as my presentation today. And this paper is, unfortunately, not one that I wrote. It was, in fact, published in 2016 by biologist and systematist Florian Boucher and a physicist, Vincent Demery, both from France. The article describes a model for an evolutionary process. We could call it sort of Brownian motion with reflective bounds, and provides mathematical details on how to fit this model to observe data for a phylogeny. I have pictures of them here. Sorry, I just pulled those off the internet. So before continuing, I should say, though, that I'm not the only one to appreciate the significance, and I'm a latecomer, in fact, and that there's another great article written by Florian and several others, including Luke Harmon, Joseph Weda, that follows up on this original article published in 2016. But I also think that this one is underappreciated. Okay. So to provide some context for this article, Brownian motion, also called sort of a continuous time random walk, stochastic diffusion, is the most commonly used model for continuous trait evolution of phylogenies. This is kind of what it looks like. This is partly for some sensible reasons, like the central limit theorem, partly for mathematical convenience. This animation sort of shows an illustration of a standard sort of unbounded stochastic diffusion process on a simple 8-taxon phylogeny. The Brownian process has two parameters: the initial condition, that is the root state, and the rate, that is the rate of variance accumulation via this stochastic diffusion process through time. So Brownian motion with reflective bounds, on the other hand, behaves kind of the same way as unbounded Brownian motion when the process is far from any boundary value of the phenotype. But when the boundaries are reached, the process just kind of bounces back in, right? And this is an animation of that process. I should note that bounded Brownian motion might be a useful model for studying, say, evolutionary constraint. I would say presently, an entirely different stochastic process, Ornstein-Uhlenbeck, which is sort of a Brownian process with a rubber band, or Brownian motion with a reverting tendency, is often used to study evolution when the sort of hypothesized process is really more of a bounded process. Actually, OU and bounded Brownian motion have different dynamics. They have different equilibrium distributions and so on. Okay. So when our data have arisen via Brownian motion, or via a process that's sort of reasonably well approximated by Brownian motion, we can compute the probability density of a set of observed values at the tips of the tree just by evaluating normal distributions. So remember, we typically, I think this came up in the previous talk, we typically need to be able to do this to compute this probability, the probability of our data, if we want to do something like, say, find the set of parameter values that make this probability the highest. These would be our maximum likelihood estimates. So I'm just going to focus, for simplicity, I'm just going to focus on computing the probability for a datum, for the data value for a single taxon here, once again under the standard sort of unbounded Brownian evolution process. And this will hopefully make sense why I'm doing this here. And here I'm going to focus on this single taxon that ends with the red point there. Okay, so if the initial condition of the process is 0 and the rate of evolution sigma squared is 1.2, which in fact they were for this simulation, then the probability density of any taxon is normal, right, with a variance that's equal to that sigma squared parameter multiplied by the total depth of time separating the taxon from the root, and the expected value is equal to the root state. So to compute the probability density of our observed datum, for the red taxon on the tree, this merely involves evaluating the normal density function for which an analytic form exists, as you guys all know, at the observed value for that taxon. Now, of course, in practice, our observations for the set of 8 taxa in our tree are non-independent one from the other. This means that their probability density is actually multivariate normal with a covariance structure that's determined by the tree, right, by the proportion of shared ancestry between each pair of taxa. Visualizing a high-dimensional multivariate normal distribution isn't straightforward, but computing the density value is trivial and can be done quickly by computers. And so that's for unbounded Brownian motion. Even though this process of bounded Brownian motion seems like an incredibly simple extension of sort of regular Brownian motion, it's trivial to simulate. The model is much more difficult to fit to data. And to see why, we can sort of first consider just evaluating the probability of the tip value for the same red taxon, but this time given our particular value of sigma squared, the starting value and the bounds under the process. Okay, so now to see how we might hypothetically compute this probability, we can start with a normal density because this would be the probability density if the process was unbounded. And I'm going to take this opportunity to remind you that even though I'm only showing this density for part of its range, the normal density function actually goes from negative to positive infinity. And so to compute the probability of our observed value for the red taxon under a bounded process, we need to proceed to fold this density. We can start by doing that first at the upper bounds, and then at the lower bound. And then we see that the upper bound tail folded backwards is sticking out the lower bound, and so we need to fold that back in, and then we fold the other one, and so on. And actually, we have to do this ad infinitum because, remember, our unbounded normal density is non-zero from negative infinity to positive infinity. And so to compute the total probability of our datum, we have to sum over all these infinite layers of the normal density function folded one on top of the other, et cetera. And of course, if this wasn't bad enough, we already haven't discussed the fact that, of course, our data that arise on a phylogeny are non-independent. So this isn't a univariate normal density. We're trying to fold its multivariate one. So that's not going to happen, basically. So what to do? So what Boucher and Demery realized is that stochastic diffusion could be approximated by an ordered and symmetric discrete state Markov chain. This is key. So the discrete state Markov chain is just the standard way that a discretely valued character is typically modeled in phylogenetics in lots of fields. And saying that it's ordered means that the changes are not allowed to occur between non-adjacent levels of the trait, and saying that it's symmetric means that the back and forth rates of change between any pair of adjacent levels are the same, and saying it's equal means that they're all the same for all pairs, okay? And then in the limit, as the number of states increases towards infinity, this approximation converges on exactness. So it becomes exactly Brownian in the limit as the number of states increases. And so what we're going to do is we're actually going to discretize our phenotype space, okay, and then fit a continuous time Markov chain to our discretized state under this constraint that the process is an ordered and symmetric process with equal rates. And this becomes increasingly exact. In other words, I characterize it as approximation, or the discrete approximation, but it becomes increasingly exact. It's asymptotically exact as the number of states in this chain increases. So when this was first explained to me, I didn't believe it. Luke can attest to that, and so can Mike. But this is kind of one of those things where once you understand it, so obviously you can't believe you didn't understand it before. And as soon as I learned it, I remember saying something like, well, of course, this is probably obvious to anyone who really studies like probability or stochastic processes. And sure enough, I'm reading this book now called Thinking Probabilistically, and it's like in chapter 2. It's also a bit of a humbling reminder that we're stumbling around in the dark, at least I am sometimes, maybe by this random diffusion. Okay. So just as a reminder, this is what Brownian evolution looks like. In this case, 200 iterations of Brownian motion for 100 units of time. And so the histogram on the right side of the plot shows the distribution of trait values across these 200 iterations, which is normal. And now for comparison here, I have Brownian process and an ordered symmetric equal rates Markov chain in which the states of the chain are quantitative values of the phenotype. And so I'm asking you, can you tell which is which? And then I'm actually going to tell you that I'm just kidding because both are realizations of the ordered Markov chain. And so to see how we might compute the probability under this model, I'm going to use a very simple example. And this is for Luke Harmon's favorite tree, the two-taxon tree. So to compute the probability density of our observations across the tips of the tree, we just need to evaluate our normal density. This gives us and then take the product. This gives us a total probability density of 0.0198 in this case. And now to make the same evaluation using our discrete approximation, we need to first build a value of the matrix, the transition matrix Q, and then populate it with transition rates based on our sigma squared and interval width of discretization, and then we simply exponentiate and multiply by elapsed time. And so this is what we get. To then compute the estimate of the probability density, we divide the product of the probabilities, because we have a probability distribution as opposed to a density, by the interval width. Divide the product of the probabilities by the interval width raised to the nth power for n taxa. This is just doing the reverse of integration. So instead of obtaining a probability by integrating the density on an interval, we're getting the density by dividing the probability by the interval width. And so this is for 21 bins. We get a density of 0.01859, and for 51, 0.0183. For 101, we get 0.021. I don't know why. And then for 201, we get 0.0197. The point is that in general, we're converging on the true known density here. And this is generally going to happen. And so how does this help us to fit a bounded Brownian motion model? Well, even as I described sort of great difficulty in obtaining the probability density in our continuous space, there's no trouble at all in finding the probability distribution in a discretized space with bounds because our continuous time Markov chain can just bounce off the limits of the space, okay? All we need to do is just to design our Q matrix in such a way that the boundary is reached, the process bounces back in at the same rate, okay? And there's no problem, and it works. And so these are values of sigma squared, the rate parameter, and on the left I'm showing simulated under bounded Brownian motion estimated with bounds. Predictably, we underestimate it, particularly as the value of sigma squared increases relative to a fixed sort of set of bounds. And then on the right I show the estimated value of sigma squared compared to its generated value when estimated using the discrete approximation as implemented in PHYTools. And it also is important, and this is an example of how it can be important. These are just measures of the accuracy of ancestral state reconstruction when we generate data under bounded Brownian motion and then estimate using either unbounded, assuming either unbounded Brownian motion or bounded Brownian motion. In my last 1 minute or 2 minutes, I'm going to just describe another couple of applications because the key thing is that when I understood this, I realized that it could be applied to a bunch of other things. And one of the things that it can be applied to is the threshold model. This is a model in which the underlying evolution is in a continuous character called liability. And then the thing that we observe is the discrete character. And as soon as liability crosses the threshold, the discrete character changes in state. This is just an example of how it can impact the accuracy of ancestral state reconstruction to use the fit our model using the threshold model as opposed to an ordered MK process, which actually in some ways is kind of similar to an ordered threshold process. And the last and kind of more interesting potentially application that I've developed for this is one for the study of the joint evolution of a discrete and continuous character in which the discrete character influences the rate of evolution of the continuous character. And in this case, what so doing allows us to do, basically we're just going to expand our Q matrix such that we have the rates of evolution within each level of our discrete character and then the rates of transition between the levels of the discrete character. And modeling these things together improves the accuracy not only of the estimates of the evolutionary rate for the continuous character, but also of the rate of transition for the discrete character, which makes sense because the continuous character in that case would contain information about the evolutionary history of the discrete character. We can also fit hidden rate models and so on, and a lot more that I'm hoping to work on in the future. So I'm really excited about this, and thanks for your attention.
Hi, everybody. My name is Jeff. Thanks for coming to my talk. Today I'm going to be talking about chromosome number evolution in a clade in the sunflower family called the Macarantherinae. Okay, so a major known driver of chromosome number variation in plants, especially angiosperms, is whole genome duplication, which, as the name implies, is going to result in a doubling of chromosome number. And what this figure from my colleague Michael McKibben shows is that if you trace the number of whole genome duplications from the root of the angiosperm phylogeny to any given tip, you're going to find you're going to find an average of 3 to 4 ancient whole genome duplications. So almost every angiosperm lineage has experienced multiple rounds of whole genome duplication in its history. So if you're starting from a base chromosome number of around 7, and you have all of these successive duplications without chromosome number reduction following each one of those duplications, we might expect angiosperms to have chromosome numbers well into the triple digits, right? But that's, of course, not what we see. So this figure shows the haploid chromosome number distributions in angiosperms, animals, and ferns. So angiosperms are shown in blue, and what you can see is that the vast majority of angiosperms have somewhere around 9 or 12 chromosomes. And interestingly, this pattern matches very closely to what we see in animals, shown in orange. even though animals do not experience whole genome duplication with nearly the same frequency as angiosperms. So clearly something is resulting in chromosome number reduction following angiosperms, but we don't really know what drives this process. So to gain some insight into drivers of chromosome number reduction, I study this clade, the Macarantheraceae. Like I said, it's in the sunflower family, Asteraceae, in the tribe Asteriae. And it is unusual for its very low chromosome number distributions. So you can see in this plot on the right that, that all of the taxa in this clade have haploid chromosome numbers of 6 or lower, except for a few presumably polyploid species with like 12 or 10 or 18. And this includes one especially interesting guy. This is the first plant discovered with only 2 chromosomes, which is the lowest that we know of in any plant. The Macarantheraceae has about 200 species with centers of diversity in the Southwest, so the Sonoran, Chihuahuan, and Mojave deserts, as well as the California floristic province. And not unlike a lot of clades that have, that occupy these kind of xeric habitats, there's an unusually high proportion of annuals. There's about 28% of these species are annuals. And this is interesting because it has long been hypothesized that reduced chromosome number might be adaptive in annual species, particularly obligate outcrossing species that occupy these kinds of unstable habitats, like desert habitats. So in my talk, I'm going to be addressing 2 main questions. The first is just how have the Macarantheraceae attained such low chromosome numbers? For example, do they exhibit higher rates of chromosome number reduction, also called descending disploidy, than their relatives? And if so, is rapid descending disploidy associated with the high proportion of annual species, as we might expect from that hypothesis? Okay, so to address the first question, I use a model called Revell. This is a maximum likelihood framework for estimating rates of chromosome number evolution across phylogeny. And this figure here shows the types of chromosome number transitions that are included in the model. So first, an increase by a single chromosome number is called ascending disploidy. This occurs by fission. A decrease by 1 is called descending disploidy, occurring by fusion. I talked about whole genome duplication, which doubles chromosome number. I'm just going to be referring to that as duplication going forward. We have this other rate called demi-duplication, which accounts for things like hexaploids. So this would multiply the haploid chromosome number by 1.5. And then we have base number increase, which for our purposes today, we can kind of just skip over this. And Revell was recently updated to allow rate heterogeneity across the given tree, which is very useful for detecting rate shifts, like the kind that I suspect may happen in the Macarantheraceae. Okay, so this figure shows what we might, a couple of patterns that we might expect to see. So given a background rate of chromosome number evolution shown at the top, so for example, these might be the rates estimated for the whole family, Asteraceae, or for the tribe, Asteriae. In order to get to reduced chromosome number, we might see a pattern, sort of like something on the right, the bottom right, where descending disploidy, shown in green, rapidly accelerates, while all of these other rates, which are all just different mechanisms of chromosome number increase, might remain relatively stable. Right? Or we might see something like what we have on the left, where descending disploidy happens, but remains relatively stable, and then we just see an attenuation of all of those rates of increase. So in order to estimate these rates in terms of events per million years, I generated a time-calibrated phylogeny in Revell. I used ITS and ETS sequences and a single fossil calibration at the root. And this is the tree. This is the resulting tree. So you can see the Macarantheraceae are up there at the top in the sort of purplish and reddish hues. And then all of these other taxa are in the tribe Asteriae. These are mostly North American species. And this resulted in an estimated age for the group of 4.15 million years, placing it in the Pliocene epoch. So using this phylogeny, I tested the fit of multiple Revell models, including rate shifts. And the best fitting model included 2 rate shifts. So one as we expected at the base of the macroantherae, and that's shown in green here. And then there was another rate shift at the base of a related tribe called the Symphyotrichini, shown in blue. And so then the sort of background rate applies to all of these yellow, golden branches. And so this results in 3 distinct rate regimes across the tree. And the gray bars that you see on the tip there show the haploid chromosome numbers for each of these taxa. So if we just sort of quickly examine those chromosome numbers, you can get an idea as to why there are these 3 distinct regimes. So at the base of the tree, we start with 9 chromosomes. In that background group, for the most part, it stays at 9 chromosomes. And then these spikes represent whole genome duplications. If we then look at the Symphyotrichini in blue, we see a lot more variation in general. Those numbers are kind of going up and down. We have polyploid changes, we have disploid changes. And then when we get down to the macroantherae, chromosome number drops down to 6, and we have much lower chromosome numbers overall. So 3 distinct patterns. And these were the rates estimated for each of those 3 groups. So first of all, as kind of expected based on those numbers, those chromosome numbers, the background group overall just has very low rates of chromosome number evolution. There's very little change going on. And then if we compare that to the macroantherae there at the bottom, we do see a sharp increase in the rate of descending disploidy, shown in green, as well as a more moderate increase in the rate of duplication. So interestingly, if we compare that to the Symphyotrichini, which overall does not have as low numbers as the macroantherae, the Symphyotrichini actually has a more rapid rate of descending disploidy. But it also has much higher rates of gains, right, of ascending disploidy and of whole genome duplication. And this is actually very similar to the pattern that we see in the family overall. So these rates were estimated by Xing Zhan and colleagues. And you can see that some of the rates are kind of slower than the Symphyotrichini, but overall the pattern is very similar, right? Descending disploidy is very rapid, but so are all of these rates of chromosome number increase. So the macroantherae really stands out for having just a high rate of descending disploidy with much lower rates of chromosome number increases. So going back to our sort of predictions, the rates that we see in the macroantherae really seem to fit most closely with this one on the bottom left. I want to emphasize that this is a relatively rapid rate of descending disploidy for angiosperms, but it has to be coupled with these low rates of chromosome number increase to get to that very low chromosome number distribution that we see. Okay, so to test the second question about the role of annual species in the group, I used a model called bichrome, which is very similar to Revell, except that it allows for distinct rates based on a given trait. So in this case, I'm testing for different rates of chromosome number evolution in annuals versus perennials, specifically testing for a higher rate of descending disploidy in annual species. So I fit 2 models. We have a full model where all the rates for annuals and perennials are independent, and then a reduced model which just has equal rates of descending disploidy for both of these groups. The marginal log likelihood values for each of these models indicate moderate support for the full model. So that means distinct rates for the 2 groups. And that support comes from this kappa value, which is just the difference between the marginal log likelihoods, and a value over 1 suggests moderate support. Not super strong support, but moderate support. And then what do we actually see for those rates of descending disploidy? It seems to be over 4 times faster in annuals than it is in perennials. So, which is very cool, right? So to return to the questions that I started with, how has this group attained such low chromosome numbers? They do exhibit higher rates of descending disploidy than that background group that I showed you, but that high rate of descending disploidy has to be coupled with attenuated rates of gains to get to the low numbers that we see. And then as to whether rapid descending disploidy is associated with the high proportion of annuals in the clade, yes, it definitely appears to be associated with that. The descending disploidy was much more rapid in annuals. So the more annuals you have in a given group, you might expect to see a lower chromosome number distribution in general. So just to put this in kind of a wider context of chromosome number evolution in angiosperms, This might give us some insight into what is driving chromosome number reduction following whole genome duplication as well. So this figure from Wood et al. Shows the estimated incidence of polyploidy in any given genus based on that genus's base chromosome number. And so the basic idea of this figure is that the higher the chromosome number of that genus, the lower the rates of polyploidy in that genus. And since whole genome duplication drives chromosome numbers up, basically what that means is that after a whole genome duplication, you're not likely to have another whole genome duplication right away. Following a whole genome duplication, rates of chromosome number increase are likely to be attenuated. So we could see that a similar mechanism where chromosome number reduction following whole genome duplication results from a rapid rate of descending disploidy accompanied by restricted rates of whole genome duplication and probably ascending disploidy as well. I finished so much faster than I was expecting. So I want to thank the Barker Lab, especially my advisor, Mike Barker, and University of Arizona EUB department, the Graduate and Professional Student Council that funded my trip to this conference. Also, shame on me, I did not include an undergrad who helped me with this project, Cole Fields, in this acknowledgements, and also Rosanna Zenil-Ferguson for her help with the bichrome model. Any questions? Yeah.
So if you compare the reduced model and the full model for the polyploidy rate, what happened? Is it that is the same polyploidy animal perennial but more reduction, or is it less, even less polyploidy? What is the combination of those two?
Oh, that's such a good question, and I can't recall off the top of my head. I think the rates of duplication were comparable, actually, in the two, but let me double check that out.
So that's like, contrary to what Steven said, I think he expected more polyploidy in animals than perennials. Yeah.
Is that the case?
Yeah, I think so.
Oh, okay. Well, that's very interesting.
Yeah. All right.
Yeah.
Can I ask the undergraduate question at the back? Hello.
Sorry. Oh, hi. Yeah.
Hi. Can I ask the undergraduate question? What's the adaptive reasoning for having low chromosome numbers in annual difficult environments?
Sure. It's thought to be associated with restricted recombination. So Stebbins and Grant talked about plants that occupy pioneer habitats or like disturbed habitats, where if you have a disturbed area, open area, and then you have a small number of colonizers, if you've already built up an adaptive combination of alleles, then it would be ideal for them to be able to rapidly expand their population by just kind of recreating that same phenotype over and over. Yeah.
So that signifies that we should see less pressure to reduce chromosome number in sulfurs because you're already suppressing the reactive combination.
Yeah, exactly. So originally, that's why Stebbins hypothesized that it would be specifically it would affect outcrossers, and he cited some data that showed that sulfurs tend to have higher chromosome numbers around 9, like what we saw in the background group.
Hi, my name is Juan Diego Rodriguez. I'm a master's student of Universidad Nacional Autónoma de México, and today I'm going to share you a little bit about evolution of sexual size dimorphism and body size evolution. So, as you may know, in animals, it's really common that males and females are different. In some species, the males are larger, but in many others, females are the larger sex. But since they have different roles in each species reproduction and have different ecologies, we shouldn't ask why they are different. We should flip the question and ask why should sexes be the same. So, as you may know, body size is a really important trait in an organism's life. It's related to a bunch of factors. Some of them are related to the way in which an organism interacts with its environment, and some others are related to the way in which an organism contributes to its species' reproductive success. So jointly, all of these factors can alter the evolutionary trajectories of the trait, meaning that the way in which a trait changes through time. And since there can be differences between males and females in these factors, we should expect the emergence of sexual size dimorphism. So the macroevolutionary patterns of SSD are related to three main forces. The first one is sexual selection, for example, through selection of bigger females, through fecundity, or bigger males, through competition and territory defense. Natural selection through niche divergence to avoid competence between sexes for resources. And one important one is the genetic correlation that exists between male body size and female body size, because sexual size dimorphism is the product of differential expression of body size in males and females. To study this phenomenon, I chose to study lizards. They're an amazing group. They have a wide range of SSD as well as a wide range of body sizes. They have a wide and amazing range of functional diversity. We have arboreal species, fossorial species, terrestrial species, and we also have available morphological data and phylogenetic data to study these organisms in a wide range of scales. There are studies about SSD evolution, but most of them are related to the allometric relationship between size and SSD. But few have been focused on the evolutionary trajectories of both body size and SSD. So my main research question is if FSD is the product of difference in the expression in body size between males and females. I think that males and females will have different evolutionary trajectories. To study that, I chose lizards, as I told you. I chose 4 families, 3 of them from the Americas, Annolidae, Liolemidae, and Phrynosomatidae, and 1 of them from Africa and Asia is Camaleonidae. Also, I chose a pooled analysis with lizards, meaning squamata without snakes, using these phylogenies. And I have to note that in the pooled analysis, I had to remove species from Tonini phylogeny that were lacking molecular information. And in Camaleonidae, I was only able to access the MCC because the posterior distribution of the trees wasn't available. The morphological data was taken from databases from Meri in 2018 and Velasco et al. In 2019. And the trait that I'm using is not Vedlecht, log transformed. And the SSD is the ratio between male body size and female body size. So what is the phylogenetic distribution of these traits? As you can see, generally, there is a bias in some groups toward bigger males, but some groups have distinct patterns of SSD. In here, we can see Furcifer, that is really male biased, and Bruchesia erypilion, there are pygmy chameleons that have subtle or absent SSD. In Liolemidae, we can see that in general, Liolemus has like a wide range of SSD, but Fimaturus, that are big species, have mostly absent or subtle SSD. And in Phrynosomatidae, it's really, really amazing that we have clades like the one that are with the arrows that are comprised mainly by female biased species. So the next question that I wanted to answer is if sexes are evolving at different paces. So these here are evolutionary rate distributions from a set of 100 trees from posterior distribution of Bayesian analysis. And I found that in most of the groups, males are evolving faster than females. But there is one exception. In Liolemidae, the evolutionary rate parameters that I found for females are higher than the one for males. And also I fitted models constraining both rates to be equal and allowing them to vary. And I found that the preferred models allowed different evolutionary rate parameter sigma squared. In here, I wanted to know if between taxa there are differences in the evolutionary rates. And I found that in Liolemidae specifically, the evolutionary rates are higher. But as you can see, the variation in this parameter distribution between trees from the posterior distribution is really wide. And I think that is maybe because of phylogenetic uncertainty, because this group exhibits a lot of introgression and hybridization that can increase phylogenetic uncertainty. Next, I wanted to know if the disparity through time accumulates differently between traits. In here, we can see male disparity through time accumulations, and is the expected under Brownian motion, and is the same in females. The curves are slightly different, but are basically the same. But when analyzing sexual size dimorphism, I found that there is not a significant decrease in disparity through time in sexual size dimorphism. As well, as you can see in Liolemide, there is a slightly increase in disparity through time. And also, we can find that near the present, there are like peaks, but you have to be suspicious when you see that, because it is possible that it is because incomplete taxon sampling. As you remember, I only had access to the chameleonide phylogeny, but the patterns that I found are the same. As you can see, male and female body size accumulation of disparity is basically the same, but you can see that there is not a decrease in disparity in SSD, and you can see like differences in the curve with the expected under Brownian motion. Also, when comparing evolutionary rate parameters of male body size evolution and female body size evolution, the male body size evolution in males in chameleonide also is bigger. So my takeaway messages is that body size evolution of males and females is partially decoupled. They evolve in similar ways, but at different paces. Also, these differences are possible because one sex is having higher evolutionary reliability, possibly by, for example, hormone effect on growth. The disparity patterns are the same because they are basically the same trait. And factors like viviparity, mating system, and ecology might be playing an important role in the evolution of sexual size dimorphism and the evolution of these differences in the evolution between males and females. Regarding SSD evolution, SSD is a species trait, meaning that it's a trait that cannot be related to an organism individually because it's a property of the species and behaves in different ways. There is not a decrease in SSD disparity through time, and I think that is maybe because SSD is oscillating between bounds that are due to the genetic correlation that exists between male and female body size. And also, I wanted to know that some clades display particular patterns of SSD. It's not homogeneous through the whole phylogeny, but some clades are special. The things that I take as future directions is study factors like habitat and environmental conditions on the evolution of SSD, study the mechanisms driving SSD bias, especially in Celoporus. This is a really promising group because we have clades with mainly female-biased species, and next to them, we have clades with mainly male-biased species, and I think that is really interesting to study the mechanisms that drive those types of shifts. Also, I think it's really important to address how it's evolving within species variation in SSD. We have like values for each species, but along a species, we can have variation in these rates. Also, I think it's really important to study SD in various ecologically relevant traits. Body size is only one of those traits, but for example, studying evolution of head shape differences or body shape differences is really important. For example, body shape can be related to the way in which females and males are using their microhabitat. Also, I wanted to study potential hard bounds to SSD because, as I told you, it's the same trait, and I think that there are like limits that are possibly like limiting, like driving evolution of SSD, and those limits can change through the evolutionary history. And also, I wanted to study the evolutionary correlations between male and female body size. Not the raw correlations, but the correlation between the speed of male body size and female body size. Finally, I wanted to thank all of the institutions that are supporting me through my master's degree. I wanted to thank my tutorial committee, and I wanted to help my lab mates, friends, and special people that are helped me during this project. And also, muchas gracias to all of you. Thank you for listening to me, and I'm open to any questions if you have one. And thank you very much.